webinar titled Cancer Diagnostics Based on Microfluidics. Before we start, let's briefly introduce ourselves first. I'm gonna go first. My name is Ujin, and I'm the second year PhD student in Professor Geo Park's lab. I'll be the moderator for today's webinar. Next, Minsa, please introduce about yourself.
Hello, my name is Min Seoba, and I'm doing a master's program in Professor Geo Park's laboratory. I'm a first presenter in today's webinar.
Taehi,
Hi, my name is Tae Kim. I am master's student in Professor Jung Kyun Jung's labs. Nice to meet you.
okay, next, Donghyun.
Hello everyone, I'm Donghyun Han and I'm in Professor Jae-gyun Park's lab and I'm the first panelist of this biofusion seminar. Thank you.
Okay, thank you. Next, Eunsil.
Hi, my name is Eunsil Ryu. I'm second panelist of this presentation, and I'm a PhD student in Professor Kim Jong's lab.
Okay, lastly, Kibum.
Hello, I'm Kibum Kim from Biophotonics Lab, Kyunjang, professor.
Okay, then we will begin our seminar. Minso will briefly present to you guys about the topic for today, which is about cancer diagnostics based on microfluidics. Minso, please go ahead.
Hello, my name is Seoba, and I will explain cancer diagnostics based on microfluidics. I focused on cancer diagnosis. First of all, cancer is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body. Cancer sometimes begins in one part of the body before spreading to other areas. This process is known as metastasis. There are more than 200 different types of cancer, and each is diagnosed and treated in a particular way. Cancers diagnosed in an early stage that are not too large and have not spread more likely to be treated successfully. But as the cancer spreads, effective treatment becomes more difficult and the likelihood of survival is generally much lower. Therefore, early cancer diagnosis is important. Then I will explain about the cancer diagnosis. Cancer can be detected by physical exam or imaging test or biopsy. In biopsy, there are two types of biopsies, tissue biopsy and liquid biopsy. Tissue biopsies are the gold standard source for tumor molecular analysis used to confirm, diagnose, and classify tumor types, as well as to guide therapies. However, performing a tumor biopsy in a patient is an invasive procedure and often challenging. In some cases, the size of the tumor and the amount of sample extracted from it is not sufficient to perform the various molecular tests needed for appropriate diagnosis. On the other hand, in liquid biopsy, tumor shed several components that travel in the bloodstream throughout the body. There are cells breaking of the tumor, DNA, RNA, and proteins released by apoptotic or necrotic tumor cells, or proteins and exosomes secreted by tumor cells. These blood-based tumor biomarkers can provide similar information as a tissue biopsy, possibly pinpoint the identity of cancer origin. And it can also be used to monitor cancer progression or evaluate therapeutic efficiency. Therefore, we can perform liquid biopsy using patients' blood with less invasive and less expensive. In addition, with liquid biopsy, it can be collected at different time points during the therapy easily. For liquid biopsy, there are some biomarkers in our body. First of all, circulating tumor cells, CTCs. Tumor cells are said from primary and metastatic tumor site and travel through the bloodstream as single cells or clusters of tumor cells. Most patients with metastatic cancer have fewer than 10 CTCs in 1 milliliter of blood. The goal of CTC technology is to isolate single CTCs in sufficient numbers with high purity from large volume of blood and low shear stress to minimize cell damage. It is present in small amount, so it requires extraction and cultivation procedure. And cells can be easily damaged. It is disadvantage. Next, circulating tumor DNA, ctDNA, and other nucleic acid fragments are released from dying cells and possibly by active secretion. Because ctDNA may reflect the entire tumor genome, it has a potential clinical utility. It is now regarded as a highly specific marker and used as prognostic marker for some cancers. Using ctDNA, we can predict disease progression or treatment effect by reflecting DNA mutation. However, it can easily degradable and is not stable. And exosomes are a subset of extracellular vesicles released by tumor cells and non-malignant cells. The contents of exosomes include mitochondrial DNA, protein, mRNA, lipid, and metabolites, which can provide information on cellular identity or tissue origin. Exosomes can be found in most body fluidics, are more stable and abundant than ctDNA. Also, exosomes are present in circulation at early stage of cancers, so exosome has a potential as a cancer biomarker. However, current techniques for isolating exosomes require extensive purification steps, are laborious, and do not yield high purities. Next, tumor cells secrete abnormal levels of peptide growth factors, cytokines, and hormones that can be used as cancer biomarkers. Thus, it is critical to measure panels of proteins in parallel from a few microliter of whole blood. The detection of proteins and metabolites in microfluidics is one of the most developed and mature technology of the field, reaching successful commercialization. This picture shows affinity CTC selection method. Positive CTC selection is based on the capture of tumor cells using specific surface antigen selection and the exclusion of normal cells. Epithelial cell adhesion molecule is a widely used marker among all of the immunoaffinity-based positive selection approaches. The positive selection of CTCs can be achieved by either coating antibodies onto the reaction channel surface or by artificially adding antibodies conjugated to microparticles. Then the captured CTC samples can be stained with fluorescence marker for detection or undergoing a genomic analysis. However, the application of traditional microfluidic channels is resisted by the small surface area for ligating antibodies, thus resulting in a limited capture capacity. In order to overcome this limitation, various kinds of microfluidic channels have been designed to provide more interactions between the CTCs and channel surface. Generally, immunoaffinity-based separation methods have a high specificity and sensitivity. Although not employed in the clinic yet, microfluidic systems are likely to become a key technology for cancer diagnostics and prognostics. Microfluidic devices have been developed for the analysis of various biomarkers, including CTC, ctDNA, exosome, and proteins, primarily in liquid biopsy. Therefore, microfluidic technological advances that are used in cancer diagnosis, prognosis, and to monitor its progression and recurrence that will likely lead to personalized therapies. In some cases, integrated microfluidic technology coupled with biosensors are proving to be more sensitive and precise in detection of cancer biomarkers than conventional assays. Thanks for listening.
Okay, second presenter Tae will introduce about microfluidic device.
Hello, this is Tae Kim. I am second presenter. I'm going to give you general introduction to microfluidics on the subject of cancer diagnostics based on the microfluidics. The content is from the introduction to microfluidics. I will explain the comparisons, advantages, principle, type of the material. I will also explain the application that can be used. First, the concept of the microfluidics. Microfluidics is a field that studies of the flow of fluid in the chamber or tunnel inside the microchannel. The fluid used here is usually used in a very small amount, and the unit is usually femtometer. There are three types of microfluidics. Continuous flow refer to the continuous flow in a channel and easy to fabrication and operated. But the sample consumption is higher than all other systems. Drumnet based is more move drumnet using the flow of the fluid in the channel. This is easy to fabrication or operate, but the difficulty in control each drumnet. Digital is the observe the move the liquid drumnet due to the electrode. It has low sample consumption and good probability. However, the manufacturing process has a complex. It is a disadvantage. There are several advantages of microfluidics. Among them, I can think of a few representative. The first is cost reduction. This means that it can be analyzed with a small amount of sample and chemical. Second is save time because many experiments can be performed inside a small chip. Finally, it is possible to make a miniaturized equipment capable of point of diagnosis using this. Microfluidic chips is made using microfluidics. These chips are networked inside the small chip. In addition, it is possible to implement on all the procedure occur into the library according to the composition inside the chip. In general, it is possible to implement all the action involved in sample analysis inside the microfluidic chips. Treatment is possible from sample collection and culture and pre-treatment. It is also possible to select a desired sample to react and separate it. Therefore, it is possible to detect desired material using the sample, thus the complement. Its analysis time is usually very short, and technology is that can be applied to all field required chemical analysis. So let's take a look at the type of microfluidics. Microfluidics are there are first digital microfluidics can manipulate drumlet on the substrate using the electrowetting. This makes it possible to create a drumlet by parameter and move them in the desired direction. In addition, it can be used important tool in the field of the drug delivery and biosensing. Optofluidics is a new science that combines the field of microphotonics and optics and microfluidics. It is integrated and operated with an optical device that creates high precision and flexibility by utilizing characteristics for the light and the fluid. Also, it is high applicable to biosensor and optical switches. Acoustic fluidics is the study and manipulation of the acoustic wave using the ultrasonic waves in microfluidic channel. Ultrasonic waves can manipulate micro and nanoliter scale fluid and particles in the fluid. Therefore, when the sound ultrasonic waves are applied to microfluidic channel, complex and unordered particles in the channel specific arrangement or the selective separation in the possible. Microfluidics has undergone a number of material changes since the development of technology. When the technology was first used, silicon is used a lot, but the high cost and optical sensing could not be reached. After that, glass and polymer were used. However, glass has disadvantage that is the high cost. So the commercially available material is polymer. Also, the material that has been drawing attention recently is paper. Paper has the advantage of being very good in terms of cost, using and transport, and compatible with the biological samples. There are several possible applications for the microfluidics in the biotechnology. For the disease diagnosis, it can be used in clinical trial and point of care. It can be also be used the drug delivery and development. It can be used in industrial and environment field to know the degree of bacteria or contamination in the water or food. Thank you for listening.
Okay, now we will move on to the panel discussion. Our first panelist would be Donghyun. Donghyun, please start.
Hello everyone, this is Han Donghyun, and I'll present separation of cancer diagnostic biomarkers based on microfluidics. Before delving into the separation of cancer diagnostic biomarkers,
저 화면 공유가 잘못된 것 같아요. PPT.
네, 발표자 모드로 보이고 있어요.
발표장으로 어떻게 하는지 알아? Sorry. 그 발표자 모드 어떻게 하는지 혹시 아시나요?
일단 프레젠테이션 모드로 가서 그 프레젠테이션 모드 눌러봐요. 프레젠테이션 모드 오른쪽 아래 여기 오른쪽 아래 밑에 여기 그 다음에 거기서 디스플레이 세팅 맨 왼쪽에 디스플레이 세팅에서 그 위에 거기 한번 위에 거 눌러봐.
네. 이제 제대로 보이시나요?
잘 보여요.
죄송합니다. 다시 시작할게요. Hello everyone, this is Han Donghyun, and I'll present separation of cancer diagnostic biomarkers based on microfluidics. Before delving into the separation of cancer diagnostic biomarkers, I will briefly demonstrate the overall concept of the cancer diagnostics based on microfluidics. As previously mentioned, cancer is one of the major issues the human race is still struggling with. Appropriate and accurate cancer diagnostics is crucial to improve overall health, welfare, and human life. Conventionally, tissue biopsy has been used as a major cancer diagnostics tool. Tissue biopsy is an examination via sample of tissue taken from body. However, the test takes a lot of time limited to localized sampling embodies pain and risk, and it is invasive to patients. Due to the limitations of tissue biopsy, liquid biopsy has been highlighted to overcome such previously mentioned issues. Liquid biopsy is an examination by a sample of blood taken from body. The test is quick, comprehensive, the sample is easily obtained, and minimally invasive. Due to such advantages, liquid biopsy has been regarded as next-generation promising diagnostic tool. However, liquid biopsy also has a limitation in sample sensitivity due to its intrinsic low concentration of target biomarkers. In order to resolve such issue, microfluidics, which is science or technology that deals with small amounts of fluids, has been incorporated with liquid biopsy so as to develop microfluidic diagnostic platform that requires small volume of sample, enables quick and efficient reaction, and that is portable. There are three main biomarkers in cancer diagnostics: circulating tumor cell, exosome, and circulating tumor DNA. And I'll introduce how each biomarker is separated from patient sample in microfluidic device. While CTC is among the most promising biomarker of cancer, CTCs have very intrinsic low concentration in blood. Only 1 to 3 CTCs exist in 1 milliliters of blood sample. Such low concentration brings a significant challenge in separating CTCs from sample blood. One of the main methods to separate CTCs is immunoaffinity-based isolation with antibodies or magnetic BiS to capture CTCs. Target-specific surface markers are used to label CTCs and separate CTCs by pooling or capturing based on their design structure of the microfluidic device. Examples of the immunoaffinity-based isolation microfluidic devices are herringbone chip, CTC-I chip, and OncoCEE chips. These are the exemplary devices that are used to separate CTCs based on the immunoaffinity. Another representative examples of separating CTCs is use of biophysical property-based isolation. CTCs have their own density, size, deformability, and dielectrophoretic properties, and many researches have been conducted to separate CTCs based on the specific properties from patient sample blood. One of the examples is vortex chip that induces vortex into microfluidic flow to allow CTCs to experience inertial forces. Due to the inertial forces that are experienced by the vortex flow, the different sizes of particles are separated, and targeted CTCs can be separated from RBCs and WBCs. Circulating tumor DNA, or ctDNA, is another important biomarker that is used for cancer diagnostics. Various methods exist in separating ctDNA from blood in microfluidic platforms, and one of the examples method is magnetic isolation. For this separation, magnetism is used to selectively isolate CTCs by a magnetic bead coated with oligonucleotide probes, magnetic field, and magnetic probes. Another method is filtration-based isolation. Researchers have conducted to separate ctDNAs based on their biophysical properties, such as electrostatic attraction. One of the examples is physical traps made by a vertical arrays polymer coated with gold nanowires. Last but not least, exosome is another significant biomarker for cancer diagnostics. A variety of methods have been implemented to isolate exosomes from blood. Since exosomes are relatively much bigger than CTCs or ctDNAs, size-dependent isolation has been widely utilized in microfluidic platform. For instance, viscoelastic flow was induced to influence the position of exosomes, and double filtration was used to separate the exosomes based on their size. Also, electric field-based isolation has been used to separate exosomes. One of the researchers has used AC for the separation of exosomes. This research has applied AC electrohydrodynamics to effectively separate exosomes and increase the sensitivity of exosomes from EVs and MVs, which are extracellular vesicles and vesicles. And this is the end of the separation of cancer biomarkers based on microfluidics. Thank you for listening.
Next, Winshil will be our second panelist. Winshil, please start.
Hi, I'm second panelist. I'm discuss about on-chip genetic material extraction from the liquid biopsy. For cancer diagnosis, liquid biopsy in blood was conducted using microfluidic channel-based site separation or surface immunoaffinity method. But it is difficult to infer the exact type of states of cancer using only site or surface information. To solve this problem, recently the development of a microfluidic chip capable of not only extracting liquid biopsy from blood, but also performing a bioanalytical platform in underway. Since a liquid biopsy contains genetic material reflect the state of the released cells, it is possible to diagnose cancer and check the status. In order to acquire the genetic material in the liquid biopsy, it is necessary to remove the phospholipid bilayer. There are various methods, but currently chemical, mechanical, and electrical lysis are applied to extract genetic material in liquid biopsy based on microfluidic channels. Chemical lysis is a method of removing cell membranes by mixing reagents such as lysis buffer or detergent. Mechanical lysis is a method of bursting the cell membrane by a sharp tools or external force. The electrical lysis is the method in which the bilayer is ruptured by exposure to an electric field stronger than the transmembrane potential, and the genetic material is subsequently released. First, the chemical-based lysis chip is a method to obtain protein or microRNAs by capturing exogens on a magnetic bead and adding lysis buffer as follows. These chips require several inlets for lysis buffer injection except sample injection. In addition, as in the upper right chip, if microRNA-based PCR is performed after lysis, an additional outlet for removal is required. Because the lysis buffer inhibits PCR performance, the lower right side is a time image of the exogen attached to the magnetic bead. When the chemical lysis was performed, it can be seen that the exogen existing near the magnetic bead disappeared. Surface acoustic waves are Rayleigh waves generated on the surface of a piezoelectric crystal by alternating current applied through an interdigitated electrode transducer. When the surface acoustic wave interacts with a bulk liquid droplet, scattered sound waves produce an acoustic pressure in the liquid bulk. This acoustic pressure affects the phospholipid bilayer. As a result of confirming the exogen particle size after surface acoustic wave treatment, it can be seen that the particle size is smaller than the existing exogen size. In addition, the surface acoustic wave operating voltage and exposure time increase, the concentration of nucleic acid increases. Electrical lysis induces a strong electric field by fabricating a symmetric electrode on a chip and performs cell lysis. When exosomes or cells are placed in an electric field stronger than the transmembrane potential, the morphology changes and substances are released as shown in the picture below left. However, when a strong voltage is applied to the electrode, bubble formation and corrosion occurs in the absence of the passivation layer, which may affect the biosample. In conclusion, the development of a microfluidic chip is underway that can not only extract liquid biopsy from blood, but also perform bioanalysis platforms. There are three types of lysis methods, and each method has both advantages and disadvantages. After extracting the generic material from the liquid biopsy, a technique that can quantitatively measure the material in the chip, such as polymer chain reaction or immunoassay, is required. Thank you for listening.
Thank you. Next, Kibum will be our last panelist. Kibum, please start.
Hello, I'm Kim.
기본 안 들려. 그냥 마이크.
소리가 안 들리네요. 혹시 문제가 있는 건가요?
들리시나요?
네, 잘
들립니다.
들립니다.
네.
아, 예, 죄송합니다. 다시 하겠습니다. Hello, I'm Kim, third presenter.
프레젠테이션 모드로 하셔야 될 것 같은데.
아, 네. 죄송합니다. 다시 하겠습니다. Hello, I'm Gibom Kim, third presenter from Biophotonics Lab, Giunjung Professor. I'm going to share with you about sensing tools coupled with microfluidic chip for cancer marker detection. These days, on-site diagnosis for quick diagnosis is drawing attention, and rapid cancer diagnosis is an important way to prevent cancer from worsenings. So I'm going to introduce you to some of the ways in which you can combine the filters cancer markers with microfluid chips. I'm going to introduce you to some measurement methods and some examples that are relevant to method. Sensing tools to combine with microfluidic chips must meet some requirements. First requirement is the ease with which they can be miniaturized for a high throughput, low cost. So it's not too complicated, and we need miniaturization method that can be applied to microfluidic chips. Also, in order to enable on-site diagnosis and quick measurement, which are the advantages of microfluidic chips, rapid measurements should be possible during sensing. Finally, even if the sensing method is miniaturized, high sensitivity must be maintained. In the next slide, I will introduce the optical biosensor and electrochemical biosensor method to meet these requirements. Optical biosensors can analyze chemical and biological reactions by measuring light absorption, reflection, fluorescence, and luminescence. Optical biosensor can be classified in three ways. First, fluorescence and luminescence-based biosensors have advantages such as ease of implementation, low detection limit, high selectivity, and lots of chemical fluorescence labels available. Methods for measuring fluorescence, including PCR method using quantum dots and FRET or SAY method. Next on the SPR, the most widely used method for non-labeled labeled optical biosensors consists of the resonance oscillations simulated by incident lights at the interface of two media owing to their merits of low cost and ease of integration. SPR and localized surface plasma resonance have been introduced into the microfluidic devices. Lastly, SIRS is a scope technique that combines laser spectroscopy with the special optical properties of noble metal nanostructures with attached molecules, producing strongly increased Raman signal. This paper measured the cancer marker using quantum dot. This paper showed the using the quantum dots were employed as fluorescence signal amplifiers to improve the detection sensitivity. Secondary antibodies were conjugated to luminescence quantum dot to realize the versatile fluorescence probe that could be used for multiplex detection in both sandwich and reverse phase immunoassay. And this paper shows selective enough to be directly used in serum. These protein chips thus combine the high throughput capability of the microfluidic network with the high selectivity and multicolor imaging available. And second example is paper used the nanohole array to produce LSPR. This paper shows the label-free this SPR configuration exhibit the extraordinary optical transmission phenomenon, which is explored in the monitoring of binding events that occur on the metal surface. In this work, this paper proposed the plasma biosensor based on nanohole array built on gold film operating in collinear transmission mode by using spectral investigation for signal transduction. And breast cancer was detection using the integration device concentration of 30 nanogram per milliliter. The most distinctive features of electrochemical sensor is the ease with which they can be miniaturized for high throughput, low cost assays with rapid detection time and high sensitivity. Achieved through microfluid fabrication technology, the integration of multiple electrochemical sensors into a microfluidic chip has attracted considerable attention as an outstanding candidate for miniaturization. So compare with single analysis, the first thing that should be installed in multi-target sensing using electrochemical biosensors integration with microfluidic system is how to discriminate signals without mutual interference. And then different methods to avoid the mutual interferes were emphasized and outlined at the beginning of the review. And the first example to using microfluidic chip integrated electrochemical. This paper shows the developed disposable, easy to use electrochemical microfluidic chip combined with multiple antibodies against 6 kinds of biomarkers: CEA, CA19-9, and HP, P53, PG1, and PG2. The 6 kinds of biomarkers related to gastric cancer can be detected sensitively and synchronously in a short time. The specially designed electrode system enables cross-contamination to be avoided effectively. This methods wants better sensitivity compared with enzyme-linked immunoassay result of 394 specimens of gastric cancer sera. This paper shows the theoretical evaluation and real-time dynamic monitoring the progress of gastric cancer in near field. And last example is the high sensitivity electrochemical apta sensor for the determinations of the cancer marker. This paper shows the free electrochemical apta sensor based on ordered mesoporous carbon gold nanocomposition modified screen printed electrode has been fabricated for the detection of vascular endothelial growth factor as a tumor marker. This principle of operation of the proposed apta sensor is based on the changes in the interfacial properties of the electrical due to interaction of the immobilized aptamer at the electrode surface with tumor markers in the sample solution, which results in a change in the interfacial charge transfer resistance as detected by electrochemical impedance spectroscopy. The calibration curve for this tumor marker determination was linear over from 10 to 30 kilogram per milliliter. Thank you for listening for my presentation.
Okay, then let's begin our quick Q&A session. So if you have any questions, feel free to ask us any questions.
I have one question for maybe one of you can answer. So yeah, this microfluidics technique as well as this liquid biopsy is a very exciting field of researches which has direct impact on the clinical science, I guess. I'm just wondering, is there any like a very noticeable or very like rising startup companies which everybody knows in, for example, in worldwide or in Korea in this area? Can you introduce them or explain them a little bit? So did you hear my question?
I have an answer about this question. Maybe in this time we have not cancer diagnosis. We have already SARS-CoV-2 detection or pathogen detection. There are some laboratory market to pathogen detection.
Mm-hmm.
Some groups like Certrium has yes, and BBB or conjugate with make a antigen and antibody laboratory to detect pathogen. I know that company, but maybe in the future, most company do for detection the pathogen or cancer diagnosis to laboratory, I think.
I see. So you mean that other than pathogen like a CTC and those things, there is not that many companies yet available, right?
Yes, there's no commercial, but maybe have some overcome drawback, overcome the limitations. So we have work about this field. So I think.
So what about the Lebanon chip on a paper? I think that's very cool idea and very inexpensive way of doing diagnosis, I guess. Is any kinds of commercial companies which are producing those kinds of things in US or in Europe or in Korea?
Actually, the diagnostics based on lab on a chip technology is currently having a hard time for commercialization because of handling very small amounts of volume for the non-experts.
I see.
So there's not so many companies and researchers are struggling to working on the various methods in a way that people, even non-experts, can easily handle and handle the sample volumes, but still there are many challenges that we have to overcome. So still there are not many commercialized technologies that can be available in the current state.
I see. What's the bottleneck, by the way? So it's so when you say the large volume means, and what's the bottleneck of the commercialization then? Technical bottleneck.
There are when you are dealing with small volumes, microfluidic devices, you have to induce flows, but the flows are sometimes it can be very complex, and sometimes even non-experts do not know how to induce such complex kind of flow, and this is one of the bottlenecks for the commercialization.
Okay. Basically the flow itself. Okay. Okay.
So there is actually one question from a student, I guess. Sok Dong-yeol asked, "Thank you for your presentation. I am interested in control circuits or devices that use for the electrical control of droplets or the micro objects. Are they usually IC chips, or what kind of electrical systems are used for the controlling?" This question has been asked.
I think one of you need to answer.
I think that the controlling of the droplet using the circuit devices is very difficult, I think, because I research I searches this a variety of research papers. In papers, I just use just to show the electro circuits, measure the changes of currents or voltage. So I think passively, so I actively controlling the electrical circuits to control the drumlets is, I think, this very difficult to, I think.
Okay, seems that there are no more questions. So,
Okay.
Professor, yeah.
Yeah. Okay. Thank you very much for your nice presentation. And as you know, this is our last biofusion seminar. So we actually have a statistics so far about this. And according to the statistics, so we have actually, everybody has actually as a appropriate number of attendance. I think there's one student who has an absence because of the hospital visit, but yeah, that's actually okay. So I think we have a prepared documentation for that. So I think everybody is passed. That's a good thing. And so far, as I announced you before, that we are going to give some small gift for the first the most frequently viewed video. Okay, so but however today's we have a team for the today's presentation that need to be counted as well. So we are going to close the finite count of YouTube view by next Monday. Okay, so if you guys goes to the YouTube account of our department homepages and department channel, and then maybe during the time and next Monday around this time, our TA will actually close that view count and then announce it. So we'll get the gift certificate for the most popular presentations. Okay. Until now, according the session 7 has a most frequent most popular view so far. But for the next week, maybe you can actually have another like a new record as well. So anyway, so that's actually the last thing you need to actually I like to announce. And then other than that, I think thank you very much for your participation and excellent talk for the last semesters, and hope you also enjoy the presentations and then also your own speeches as well. Okay, thank you very much. Any question from students or presenters for me about this class, grading and etc.? Yeah, other than that, let's close our session. Thank you very much. Bye.